So hello and welcome back. This will be part 1.2C, that is in our structural overview to the immune system. We'll be talking about other cell types that are present within the lymph node. The general structure of the immune system is, as we already know, it consists of primary lymphoid organs, secondary lymphoid organs, and tertiary lymphoid organs. We're specifically going to talk about tertiary lymphoid organs, and that too specifically about lymph nodes. So in this video, we'll be talking about the following topics. We'll talk about T follicular regulatory cells, their formation and primary function. We'll talk about other follicular reticular cell types, that is perivascular and deep follicular reticular cells. We'll talk about the two dendritic cell types that are present within the T cell zone, although I'll not go into the details of their structure and function. That is conventional DC type 2 and plasmacytodendritic cells are located within the T cell zones. We'll talk about some cell types that are not usually present within the lymph node, whose presence is actually considered pathological. So the other cells within the lymph node, starting with T follicular regulatory cells, other follicular reticular cells, dendritic cells, and macrophage subsets. So we are specifically going to start with T follicular regulatory cells. Now, T follicular reticular cells, abbreviated as TFR cells, are a very interesting form of cells. Okay, so T follicular reticular cells, like T follicular helper cells, express the master transcription factor BCL6. Now, T follicular helper cells targeted against B cell derived autoantigens can obviously aberrantly cause the formation of spontaneous germinal centers, because there are a lot of B cells within the lymph node, and some of them undergo apoptosis, some of them die, and those antigens get expressed. So if you have a T follicular helper cells that recognizes such antigens, it's going to unnecessarily get activated. T follicular reticular cells specifically express TCRs that recognize these self antigens that are normally expressed by B cells, and then develop a regulatory role. So thus they prevent spontaneous germinal center formation. So as B cells undergo somatic hypermutation in the germinal center, they can just by chance develop B cell receptors with autoreactivity to self antigens, that is particularly B cell autoantigens. So that's how these autoantigens form. T follicular regulatory cells are thus a crucial third line of checks. The first and second line being the central and peripheral tolerance mechanisms, which we'll describe subsequently in our discussions on the function of the immune system. So on the development of autoreactive BCR expressing B cells. So BCL6 regulates the transcription profile of T follicular helper and T follicular regulator cells via a very interesting gene regulatory circuit that is called the repressor of repressor gene circuit. So we'll talk about these circuits again when we talk about the function of the immune system. Now, T follicular regulatory cells are a special subset of T cells in the germinal center that primarily prevent spontaneous germinal center formation, as I've just discussed. Primarily arise from two sources. The first is they initially arise from T cells that have been derived from FOXP3 T regulatory cells that arise within the thymus, and then during germinal center formation they are derived from naive CD4 plus T cells that are undergoing activation during germinal center formation. So as you can see, you've got T follicular regulatory cells which are FOXP3 positive. So they may have been FOXP3 positive right from their development in the thymus, or they may have developed it after germinal center activation within the lymph node. Now there is a complex set of cytokines that causes T follicular reticular cell formation. So we've got IL4, we've got TGF beta, but additionally we also have immunoactivatory cytokines. So CD6, CD21, interferon gamma, as well as IL2. So this is essentially T follicular reticular cells form both during the early and late stages of immune activation. Okay, so as you can see, they then inhibit T follicular helper cells and thus prevent spontaneous germinal center formation. Now, the second subset of cells that we're going to talk about are other follicular reticular cells within the lymph node. So these are twofold. That is, one is the perivascular reticular cells, and the second is what we call the deep reticular cells. Perivascular reticular cells express CD34, MADCAM, and integrin alpha 7, whereas the deep reticular cells express ER-TR7 and PDGF beta. Now, naturally, perivascular reticular cells are located around high endothelial vessels. They essentially replenish the FRC populations, other follicular reticular cell populations, in a fully developed lymph node, whereas deep reticular cells form a dense network within the deep cortex, essentially to capture antigens. So, naturally, perivascular reticular cells are called perivascular because they're located around high endothelial vessels. Okay. Now, other cell types, the third cell type we're going to talk about is the dendritic cells. We have two dendritic cell subpopulations that are localized within the T cell zone. So these are specifically within the T cell zone. This is the conventional dendritic cell type 2. Again, this is C conventional dendritic cell subtype 2. So there are two subtypes, and we have the plasmacytoid dendritic cell. These will be discussed again in detail in comparison to conventional dendritic cell type 1 cells and Langerhans cells in our discussion on dendritic cells in our functional overview of the immune system. The last cell type that I'll be talking about are the macrophage subsets that are present within the lymph node. Macrophage subsets, naturally by extension granuloma formation, is abnormal within the lymph node. Neutrophils in a large number or eosinophils in large numbers within the lymph nodes are also abnormal. So again, in pathologies we'll see that we find these abnormal cell types. But what is not abnormal, that is plasma cells and plasmablasts. This is why actually in IgG4 related disease, when you take a lymph node, diagnosing IgG4 based on presence of large number of plasmablasts, or say the plasmacytic variant of Castleman's disease based on large number of plasmablasts in the lymph node, is a little tricky, because a normal lymph node can also have significant number of plasmablasts and even plasma cells. So that's a little little odd, and then we depend on IgG4 to IgG ratios, and obviously most importantly the clinical presentation. So we'll discuss that in the context of IgG4 related disease and its differentials in our approach to mass forming lesions later. So in this topic we've discussed this screencast, we've discussed T follicular regulatory cells, their formation and primary function, that is to prevent spontaneous germinal center formation due to aberrant T follicular helper responses against B cell autoantigens. Other follicular reticular cell type that are present within the lymph node. So we described multiple types earlier, the subcapsular, medullary. Okay, so now we've described two more, that is perivascular and deep. Then we've talked about two dendritic cell subtypes that are present in the T cell zone of lymph nodes. That is the conventional DC type 2 and the plasmacytodendritic cell. Specifically, the plasmacytodendritic cell presence here becomes relevant in the context of Yakiguchi-Fujimoto disease as well as SLE lymphadenitis, and we'll get to that when we get to it. Then finally, we've talked about cells that are not typically present within the lymph node, that is your macrophages, and then by extension granulomas or neutrophils and eosinophils. So thank you for watching, and in the next video in this series, we'll be talking about the spleen as a secondary lymphoid organ and also the mucosa associated lymphoid tissues as secondary lymphoid organs. We'll use the basic principles that we've understood in the preceding three videos and apply them to these two structures. There are some macro structural differences, but the general principles are the same. So I'll see you there.